Big scary monsters.
How shall I feel? Creatures lie here, looking through the window. How shall I feel? Creatures lie here, looking through the window. How shall I feel?
A couple of months ago, Dr. Melba Ketchum founded a company called DNA Diagnostics in 1985. It's become a leader in all types of DNA testing, including human and animal forensics, human and animal paternity and parentage testing, disease diagnostics. She's been on the front lines of some pretty big DNA questions, worldwide research projects, but I don't think she's ever tackled anything quite like Sasquatch, Bigfoot. She's been trying to get this paper published for a while, has suffered slings and arrows, both from within the Bigfoot community and within the scientific community, and now she has a chance to answer back. Dr. Ketchum, great to have you back on the program.
Thank you for having me, George.
Melba, did you know it was going to be quite this tough sledding in terms of getting the paper published when you started?
Absolutely not. I thought it would be a piece of cake because the science was good, and I was naive enough to believe that, and I believe you use those words, and naive is a good one.
But you thought, well, gosh, the science is good, and the topic is so interesting. How could they possibly resist taking a look at this, an honest look at it, right?
Absolutely.
And it's been anything
floored.
but that?
I was floored.
Tell me about this journal, De Novo. I don't know about it. I know it's a relatively new journal, which is one of the criticisms that I saw online this week. Well, gosh, Melba must have made up this journal herself.
Well, that's not the case, and I've sent you documentation, haven't I, to this point?
Yes.
What happened was that after having a lot of biased review, and I mean seriously biased review, and I sent you documentation of that also, we found a young journal that was fairly new, and we were submitted our manuscript there because they were leaning more toward next-generation sequencing technologies, which is how we got our genomes. And I spoke with the editor. The editor agreed to send it out to people that, worked with next-generation sequencing. And we got back passing reviews, passed with revision, and then we made some revision and responses, and we were told that the journal would publish. And it was legitimate peer review. I have no idea who they were. I have the peer reviews. I believe I sent you the peer reviews where you could see where they accepted it from the button that was pushed at the top of the reviews. And once we received the reviews back and they agreed to publish, we were ready to send out a press release. And all of a sudden, just a few minutes before we were going live with it, I get this email, which I furnished to you. Basically, the editor is saying that his lawyer, doesn't want him publishing this. And in a further conversation, and as alluded to in the email, the lawyer will quit him if he goes ahead and publishes this. Also, that it scared him so bad that he was afraid it would ruin his career, much like it's ruined mine, because nobody wants to be associated with something that's allegedly a myth. And therefore, they backed out at the last minute. So he felt really bad, and I was able to actually acquire that journal because that was the only way I was going to be able to take those peer reviews and some legitimacy with it because, otherwise we'd have to find another journal. Well, we've already spent nearly 2 years with journals, and I'm not going to wait another 5 years to go through 100 journals to get one that will be fair.
If you could get one, if we could
even get one. So we acquired that journal and very quickly threw together a new website, new name, and published it. Now, I've had nothing to do with the publishing. Actually, the people we acquired it from, they helped make changes and put it up and had it ready to go for us, more or less. It still has some flaws, but we wanted to get the paper on out because it's been long enough.
You were hoping that just the subject is interesting enough that even if you didn't write the paper and dot all your I's and cross all your T's to everyone's specification, that they would at least think, wow, this is a novel approach. It's an interesting topic. It's earth-shattering in its importance. Let's take a look at it. Let's at least publish it, put it out there, let people read it and evaluate it. But that I read the give and take between you and a couple of these journals, and it's outrageous. I mean, it's not. It was my impression that some of the complaints from some of these people who sent in comments were from people who had not read the paper at all.
Yes, that's absolutely true, because I would come back with our response that's already in the manuscript if you will read it, basically. And that happened multiple times. And not only that, there was one review that mocked me. I think the worst thing that happened was one of the major publications wanted to have another look at the paper. They said, make all these changes, document that you've done everything they've asked you to do, and then send it back to us. Well, we did that, only they asked us for if we could get just one genome. We got 3, and they were the same species. They also, when we pulled the mito out, it was the same mitochondrial sequence as we had done individually prior to the whole genomes. I mean, it's definitive proof. And when we sent it back, one of them wouldn't even review it. And then there were 3 others that, there was no excuse. One of them still didn't read it. And the other one just didn't believe it.
The phrase that is used in your paper is the information, the data is consistent, reproducible, and novel. You had to beat around the bush last time you were here. You didn't want to spill all the beans before the paper was published. Tell us now. Give us the thumbnail sketch of what the paper says, what your findings were.
Okay. The first thing we did was when we extracted these samples, 2 labs extracted, both forensic labs extracted these samples. We washed the samples. We did all the techniques. We screened them for contamination, and there was none. We went in there and then screened for species with mammalian primers, which makes it where any animal would show up, including human. And all of our samples came back human. And this was after the hair had already been looked at by a forensic hair specialist that does primarily human but also animal. And he had said it was novel. It was not the same. It was not human hair, and it didn't match any of his animal knowns that it was tested against. So we knew we had good, clean DNA. We went forward after screening, and we started outsourcing because I knew there would be a problem with if just 1 or 2 labs did this. So we sent these samples out. Nobody knew what they were, and we sent the samples to Family Tree DNA. They specialize in mitochondrial DNA. We got 20 whole genomes, 10 partial genomes, and the rest of the samples we just screened because, I mean, you get that many that are running the same, that pretty well tells the story. Non-human hair given human sequence. And then, of course, we had some non-hair samples in there, too, but the majority of it was hair. And once we got those results, we knew that they didn't exactly look human, so we decided to go after the nuclear DNA, which the mitochondrial DNA, I might offer, is maternally related. It comes from your mother only, whereas your nuclear DNA comes from your mother and your father. So we started with the nuclear DNA, and we started getting really strange results. And normally when you run a particular primer pair, and we tried to do as many universal primers as we could, as well as human and ape-specific primers, we would line, whenever you select a particular spot you're going to amplify on the DNA, you'll get, like when you look, you've seen on TV where they have gels with bands on it, and they all are just running across at the same level. Well, that's what you get. Well, we didn't get that at all. We got weird-looking bands that were all different sizes, yet we had already species identified them as human. And so this was bizarre, and I didn't tell them once again what they had. So I get this email from one of the PhDs at the lab we outsourced nuclear testing to. We also let them do some mito testing to back up the other mito. And she said, we got this really weird fragment. It's not as long as it should be. It's like 490-something bases, and we were supposed to get 500 and some odd bases. And a base pair is just the building block of DNA, and the length of it is in base pairs. So with that said, it was a shorter one, and she blasted it in GenBank. Now, GenBank is a sequence depository that all the scientists, they put their sequences in, and then you can go compare your unknown sequence to it. Well, she blasted it, and it didn't come up matching anything that's ever been put in GenBank out of millions of sequences. And so she writes in this email, she says, what did you send me? We don't know what this is. Have you discovered a new species? I have that in an email. And so we finally had to tell them what they were testing.
And they didn't like it.
No, none of them liked it. I take it back. A family tree was very much the nicest one about it. But some of the others, we had one threaten us if we used their name, we couldn't use it.
But you paid them. You paid them. You had to use the results.
They wouldn't take our money when they found out.
Well, that's not very scientific.
And wouldn't let us use anything. We did get our pictures out of it, and that was it.
But I mean, they did the work. It's just not like Dr. Melba, crazy Dr. Melba in the closet in her house doing all the DNA testing.
No. We chose the best places in the country to do it.
Right.
I shopped around and found out who had the best reputations. They were accredited labs. They were major university labs. There were state crime labs. I mean, this was not my little lab. It was really big name labs. Texas A&M, they backed up the results. I wasn't able to use the name of. They did it, and they saw the same strangeness that the other university did.
Help me understand this, Melba. Okay, so, a lot of the emails that I've got after your last appearance here and since then, people say, I don't understand it. Why can't it just be human? And I know in some of the criticisms, and we'll get into this a little bit more later, but they'll say, well, gosh, it must be human contamination. But you have hairs, these hairs that have human and non-human DNA in them. It's not contaminated. It's not some other species. It's one species with this two kinds of DNA inside, right?
No, that's not quite right.
Okay. Right.
What you've got is, a physical hair that's about the size of a horse mane hair and wavy. And it's about 3 or 4 inches long, and it's got a natural end on it. It's never been cut. It's never been dyed. When you look at it under the microscope by a forensic hair analyst that does both human and animal, and he had a whole lot of knowns of North American animals to compare it to, it didn't fit any species that he had seen before. So he screened it first. We've got non-human hair. It's twice as big as human hair. And then we test it, and the DNA from those hairs in 100% of the cases was modern human female mitochondrial DNA. It was no animal DNA in it. It was the hair that was animal-looking.
Okay, so those who say, well, gosh, it looks to me like it's all human. There's nothing novel about it.
Well, that was fine until we went into the nuclear testing.
Right.
The whole genomes and what have you, and the different places on the DNA that we looked at for the first time. And then we found all this really strange sequence that's just not in GenBank. I mean, there'll be bits of human sequence in it, but then there'll be a lot of novel sequence, different sequence in there that's not ever been, One thing about it, at least we could use GenBank to blast with or to check the sequences against what's in GenBank without uploading them. So we were able to do that, and we have trees to that effect. A lot of the supplementary data is trees that were generated directly on GenBank.
One of the things I did not understand, and I'm asking you now to help me and us non-scientists to understand this, is you mentioned in the paper about 16 different types and you're suggesting that these individuals were not all from the same area. How do you make that determination? And am I explaining that correctly?
No, that's right. Basically, when you do mitochondrial DNA testing, it's very popular nowadays to have your mitochondrial DNA tested so you know what your origins were, where you came from.
Right.
And it's like a big tree, and it starts out in Africa, and then it moves to the Middle East, and then it starts branching into Europe and branching into Asia. And each different mutation, the mutations happen just ever so often, very slowly with mitochondrial DNA. So when it mutates and becomes a new type and makes a new branch, where that happened, usually you can kind of trace the DNA to that particular spot. So, say somebody is Native American, they would have a branch that would come from the Middle East through Asia and across the Bering Bridge into North America.
Okay.
And a particular group assignment of a letter designation, like a C or a D, for instance, are both Native American types that have originally came through Asia and across into North America.
But you can, as you talked about this before, but just to reiterate, you can weed out an animal. It's not a possum. It's not a bear. I mean, the labs you're sending it to, they can tell right away, right?
Oh, absolutely. Because the thing is, when you sequence the mitochondrial DNA, that's what forensic scientists use for a species test. Is you have, these universal primers that'll work on everything, and you sequence your DNA from them, and it's going to tell you what species you got.
Okay. Much more to come with Dr. Melba Ketchum about this groundbreaking study looking at DNA samples from purported Bigfoot. Stay with us, everyone. Much more to come on Coast to Coast. We're talking with Dr. Melba Ketchum about a groundbreaking study of DNA in purported Bigfoot creatures. And she's got about 111 samples that were analyzed in blind tests conducted by several reputable DNA labs. The results are astonishing, and yet the reaction in the scientific community kind of across the board. You got a couple of scientists who are saying, well, that's kind of interesting. I'd like to know more about that, which is the reaction you'd hope you'd get. And then you've got some people that say it's absolute nonsense. It's totally ridiculous. It can't be, and therefore it isn't. And you get the feeling that some of those folks didn't really read the paper. It certainly didn't read it with an open mind. We get back into the nuts and bolts about this study right after this on Coast to Coast AM. Welcome back. My guest is Dr. Melba Ketchum. Dr. Ketchum, where can people read the paper?
DeNovoJournal.com is the link to get to the paper, and there are links to download it from there.
Okay. And a lot of people downloading so far?
They were until we had a glitch with the website because it was put together in a hurry. So, but I think we've got that fixed now, so it'll be available for download anytime somebody wants it.
All right. You had 111 samples that were tested, collected by different researchers around the country. Dave Paulides, I know, and his North American Bigfoot folks helped you find some. Yeah. So what kind of quality control testing do you do on the samples?
On quite a few of them, we actually had the people that gathered the sample. We had their DNA to compare it to make sure that their DNA wasn't contaminating it. We also have all the lab people on file, so we knew it wasn't any of our lab people either. Like I said, we washed the samples. A lot of the samples came from either eyewitness or controlled areas where there were sightings. The Erickson Project, for instance, they actually had a study going on that was overseen by a PhD in wildlife biology. And they used, most of the collectors did use gloves and did proper procedure in actually collecting the hair. The Erickson Project collected saliva, and they would use sterile paper plates because most of your paper products, when packaged, are sterile and, or close to it, and therefore they're a good thing to use. They use paper plates with sandpaper that's glued to it to help pick up extra cells for the DNA, and they would put it in Tupperware, so you'd have to have a hand to open it. And then they would wait for them to come in and eat. The Sasquatch got used to eating, these plates, and the people that lived on the site would prepare it so they were not scared of the people particularly. They'd come in, they would eat the meal, and then they would collect the plate, immediately take it in, put it in a brand new bag, and put it in the freezer. So, there were eyewitness sightings along with research projects that were well run under the circumstances. The other good thing about the Erickson samples, especially, we have a little short video that we went ahead and released to the public of the one that's nicknamed Matilda. She's a young female sleeping in the woods. That particular family unit was very unusual in that they got so used to their people and used to being fed that they could actually come pretty close to them at times. And so that's a very good video for sample 37, which is one of the few female samples in the study.
There's a guy who, of course, became famous because he claimed to have killed a Bigfoot, maybe more than one, and he had a couple of samples of Bigfoot meat that he supposedly sent out. And even now, after your paper is out, I see you getting blasted online by people who say that either you lied about the meat or he lied, or I guess the question is that meat from a purported Bigfoot part of your study?
Yes, it is, and I'm going to give you my opinion of what I think has happened. first of all, anybody can go online and look and tell that the pictures, the two samples are different. The picture in our paper we have, it's long, wavy, mostly white hair. There's no undercoat. The sample is not dried looking. You can see fresh, pretty fresh meat on it, you know, muscle tissue and subcutaneous tissues. And that's why we turned it up to where you could see both the haired side and the fresh side. And we even had one of the complaints was that our samples were degraded. We even had histopathology done, and the cells were all intact. It was fresh. There was no bacteria to speak of on the sample. So very, very pristine sample. If you look, there's a certain blogger out there that put some pictures up about the one that they sent to Canada. Well, first of all, the DNA is different, so it's a different sample. Let's just start by saying that. If it had been the same sample, the DNA would have run the same. Second, and we have, tremendous amounts of testing. We have a whole genome on that one, whereas they took 7 months to get, just a very small section of mitochondrial DNA, among other things. So with that said, though, you can look at the picture and see that there's an undercoat on the samples that ended up in Canada. I don't, my opinion is that perhaps they got worried because these things are human to a large extent, and there could be, repercussions because of that. And I really think that's what happened, but that's just my opinion.
I'd want to reemphasize this. You mentioned it sort of in passing during our first segment together, but I think it really bears repeating, and that is that one of the labs that had one of these samples was curious about it. They sent it to something called GenLab, which is this gigantic bank of DNA information, and it didn't match anything else. Am I understating?
We did that all through the paper. We started out, we blasted the individual testing and the mitochondrial testing, and then when we did the whole genomes, once we aligned a section, we did 2.7 million bases across approximately. The three samples varied some, but approximately the largest one was 2.7 million bases, and I think it was 2.4 million bases. These samples, we could keep them in our system, but yet blast them or search against GenBank, which is your depository. When you publish a paper, you're supposed to put this into GenBank for other scientists to look at, which we attempted to do. Now, I sent you a thread of emails from one of my co-authors trying to get this done, and I'd ask you to make a comment on that.
Well, I mean, it's pretty weird that, it certainly does look like they don't want it going in there.
Right. They refused it. They wouldn't accept it because it didn't have a species name attached. So we wrote them back, basically, and then it said that, use human, use Homo sapien, but if you do, we have to have a signed written permission from the donor. I mean, good luck getting a Sasquatch to sign their DNA over to us, and a couple other rules that they had in there. So we couldn't do that. And
so you can't just put it in GenBank blind.
No, you have to fill out all this information on it.
All right.
And whenever they found out apparently what the subject matter was, they just came back, and finally we said, if you don't do it, if you don't let us put it in, we don't know what the species is because there's not a species name on it yet. We can't do this. This is the whole point of our paper, is that this is a new hominin, and we want to get these sequences in. And if if you don't get back with us, we'll just have to put it in with the paper, which is what we've done. We've provided those sequences that we used in the paper as supplementary data, for people to look at. We're transparent with this. We didn't want any problems with this, but, that's why I sent you the documentation. And people, I think that I'm just saying this stuff, but I've got the paper trail to prove it, and I hope you can acknowledge that.
I do acknowledge it, and I hope that you'll put that information, that trail of information and paperwork where other people can read it. Let me ask you this question, Melba. The idea of interspecies mating, that, we humans, we can't mate with a chimpanzee. How do you figure this can work? That somewhere 13,000 or more years ago, these two species mixed?
Well, first of all, the species identity shows it to be human. But when you look at your whole genome nuclear DNA, it is so different that it must have been just kind of a fluke to happen that way. I think that's one reason the established scientific community really has a problem with this, because they've not seen something like this, not to this degree. Now, as you know, it's been proven that we have little bits of, as a human species, that some of us have some Neanderthal in us, which is another species of human, and some of us have some Denisovan from Russia, the finger bone that was found. We have some genes left from that particular hominid. Well, this really isn't different, except it's kind of in reverse in that we're probably the lesser amount of human DNA in the nuclear part of the DNA than it is with us and the Neanderthal and Denisovans. But it's still kind of a similar scenario. But the sticking point is that the mitochondrial DNA is 100% human. The other sticking point is that it's a recent hybridization. I had a real interesting a scientist come forth and actually support our strange electron microscopy on it, where we had double-stranded DNA and then single-stranded DNA, and he was talking about that this would happen if the hybrids didn't fit real well together, because you have stretches of sequence that just didn't align with one another, and that kind of made sense. So some of the scientific community is starting to come out, and we have now sent it. We're asked if we would let some more famous, quote, scientists look at it than what I am. And we've already sent the data over to them. So there'll be some independent analysis. Even though we've done everything about twice on all of this, they're going to get their shot at it. And plus we're letting, certain scientists, really go through the paper. As long as they are open-minded, they'll see how much science is in the paper. But they have to be open-minded to start with.
Well, we'll come back to that idea, because there's a lot of them out there that certainly are not open-minded, at least the reactions I've read so far. Again, back to this interspecies mating point, it's not still going on. It happened a long time ago. You're not saying any of this is still going on, do you?
Well, there's reports, and I can't really verify them.
That's outside the bounds of the paper, right? That's outside what you're...
This is outside of the paper. This is, when you go on the Internet and you start... First of all, there's a real famous case that was in the 1890s in Russia, the story of Zana and her son Quit. And this was a young female Sasquatch, or wildman as they call them, that was captured by a mayor of a town or some official like that, and he kept her and used her to carry heavy things and what have you. He semi-domesticated her to the point that she was allowed to walk loose in town. And when they would throw parties, she would be the prize for some of the men in the community. And she ended up having 6 children. The first 2, and these were by human males in the community. The first 2, she drowned in the river because she went to wash them after she gave birth, and it was cold water. Obviously, our human nature is not as sturdy. And the next 4 they took from her immediately and were raised by townspeople. And, they looked a little different, and the skull on her son Quit was very different morphologically. So they actually did have the DNA done, and they got human mitochondrial DNA from him and from her alleged skull also. So, here we are with human DNA again. But, of course, critics say, oh, well, they were just humans. But yet, as late as the 1990s, there was a lady that was about 100 years old that was able to give a really clear eyewitness account of Zana and the way that she behaved and the way that she looked, very compelling. And then there's another case in Washington of a gentleman that was the product of a woman, a Native American woman that was abducted allegedly and came back pregnant and had a son. And this son allegedly had some really nice qualities about, he was a horse whisperer. He could talk to animals and they would understand him, it seemed. And, he had several very odd traits about him. So allegedly that is another true. And there's a lot of reports out there. Those are probably the 2 most famous. There was actually a little scientific study or a thesis done on this mating by a PhD in the Pacific Northwest.
So the Sasquatch that witnesses say they see, we know they have human DNA in them, the ones that are running around, but we're not quite sure how many humans might have Sasquatch DNA.
Yes, and that's going to be an interesting study. I've got a couple of people that think they might have such. And, if we can get funding, we'll probably go ahead and do their genomes and see if how well they align with regular human genomes.
Any Neanderthal DNA in the Sasquatch?
Not that we've seen, but, there's still, it takes years to analyze whole genomes, and 3 of them is a huge undertaking. So, it'll be a while before we know all of this. But we did some specific, we knew certain places to look for Neanderthal, and we did not find it.
The criticism you hear the most is, well, gosh, why can't we see some of this stuff? Some, at least, and we'll get into specifics, but I mean, some of these guys said, well, put out all the data. We don't see the data. Where is the raw data? And why won't she let us see it? You said you have shared this with some other independent scientists or seemingly open-minded. Is there any a plan to say a more widespread release? Put something where the genetics community of the world can take a look at it.
They have offered to help us get it in GenBank. They will put pressure on GenBank. So it will be in GenBank. Like I say, though, although it's available right now with the paper, we've got a text file of the sequences, even up to 2.7 million bases, because text is not extremely large. So we were able to use everything, put out everything in the paper in a couple of supplemental files. And, if you go to that link I sent you, you just click on the supplemental number, and the chart pops up, and you can see the data.
Am I correct in saying this is just the beginning? I mean, you don't claim that this is the end-all, be-all.
No.
You're just getting started. The sequencing numbers and yeah.
All this is to prove that they exist. That some of these came from eyewitness samples. A lot of them did, and therefore, and somebody sees it, they collect it, they send it. We get the same results across the different samples. The genomes did align in the same spot on the phylogenetic tree, so it shows that the samples came from the same species, which has not been seen before. And people are saying that they saw Sasquatch where they got these samples. So with that said, that pretty well tells you what they are.
I saw one critic who said, well, look, this is meaningless unless you've got video to go along with the sample. You should have video of Bigfoot in the forest and leaving his hair sample on the tree, and that would prove that the sample is from Bigfoot.
Well, we have the video of sample 37, Matilda sleeping in the woods. And the Erickson Project has definitive footage. But, the problem with that is, look how clear the Patterson-Gimlin film is. And they're still arguing whether it's real or not. Video will never prove it, because there's always going to be somebody making an excuse that it's not real, just like they're trying to do with the DNA. But you can't fake DNA like what we have and what we've done, where it's repeatable on a large-scale basis.
You can't fake it.
No, you can't fake that. Because if you, to me, the largest criticism is, the samples are contaminated. Well, the thing is, you have what's called, when you do the next generation sequencing, there's a quality score. And when you prepare the libraries to sequence, it's a competitive reaction. So if you have, say, 2 different types of organisms, it's going to use up your reagents quicker, and you're not going to get as much repetition of the sequences. So it will affect your quality score. And I talked to the people that make that machine, and it was 2 supervisors actually. And I asked them, I said, is there any way to tell if our sequences are contaminated? Because somebody had mentioned the Genome Center had probably contaminated them. And even though they only test human, and they were saying they contaminated with animals. So we actually had a conversation, and immediately the comeback was, well, if it's contaminated due to the competitive nature of the reaction to prepare for the sequencing, your quality scores would drop down to 40 or 50. And I said, well, what's an average one? And he says probably about 85 for just the average genome that's done on humans. And their actual printout about quality scores says 80 or above. All 3 of our genomes were 88 and better. So we were way above what the average quality score is.
Dr. Melba Ketchum is my guest, talking about her groundbreaking Bigfoot DNA study. Much more to come on this part of the conversation, including covering some of the criticisms that have been raised so far. We're talking with Dr. Melba Ketchum about her BigFoot DNA study. And, maybe 50 years from now, maybe 20 years from now, other scientists will have had a chance to study the information, and it will be a foregone conclusion. Not quite so controversial. But for now, though, it's tough for a lot of these folks to swallow it. Maybe it won't be so tough if they'd be willing to give it a fair look. We'll talk about some of the criticisms that have been leveled so far as we continue our conversation on Coast to Coast AM. Dr. Ketchum, the last time you were here, there were entreaties that you made to a Russian journal that maybe they were going to publish the paper. What happened there?
The same thing that happened with the major journals here. It's got to be contaminated. And the thing is, we have definitive proof it's not. That's what's so sad about it. But it's the easiest explanation. I can see where if you don't want it to exist, you don't believe in it, nothing's going to convince you. The only explanation would be contamination of human and unknown or animal in this case. And but like I say, with the Q30 scores being so high and the fact that we worked them up forensically, we had 2 different labs work them up, 2 different extraction techniques, screened for any contamination, and then the same extractions used all the way through. We aliquoted the samples and the DNA extractions that had already been screened, and everybody got the same extractions to use. So it's not like we did multiple times that could have gotten contaminated at some point.
I know that there was another study that was launched after yours. It looked like it was almost a race, that they saw what you were working on and at Oxford, big-name scientists who said, yeah, we're going to jump into this pool as well, and they were going after it. Where do they stand now? Do you know?
Well, they're behind us. I know that. I really don't know. I've had a request. He wants to come talk to me, but I don't know. I don't know anything about what they're doing. We don't have anything to do with that study.
How would you characterize the reaction of the scientific community? I mean, you knew it would be hostile to some extent. Is it more hostile?
Is an understatement.
Right.
I was shocked because I really thought more people out there would. I mean, there's been so much reporting in this country of sightings and thousands, literally thousands. One group has come out publicly and said they've got over 30,000 sightings in their database, and that just is the tip of the iceberg. We've had so many emails of people being really happy that this has come out, saying they're vindicated now, that they can come out and say, yes, I've seen one, and they wouldn't do it before because they were afraid of getting mocked and made fun of. And I'm guilty of that. I did that to a friend of mine that had a sighting back in 95. I didn't believe her. I basically, made fun of her over it and said, I don't know what you saw, but, it wasn't, because I didn't believe they existed.
I guess one good reaction could be that you're going to get more samples.
You know what? At this point, we can take more samples, but most of with the 3 genomes, that's going to be the main thrust of any forward movement because, we've got a bunch of genes that have now been aligned, and we're looking at those, and there'll be more papers maybe, assuming that somebody will actually read them. I think we're going to be vindicated, though, because we actually did have, like I say, a group come forward and when I say famous scientists, our group is famous in our own way because we're forensic scientists. We all testify in court. I've been real active with, in the past with the International Society for Animal Genetics. I've chaired, several international committees. So, it's not like I'm unknown, but forensics and animal genetics it's almost like there's an invisible wall between them and human genetics. And the people that are looking at this are human geneticists for the most part. But now we've had some human geneticists come forward that, are very well known that, like I say, have our data, and they're looking at it now. I don't know how long it'll take. I wouldn't think it'd take too terribly long. There's a blog site. I looked it up in preparing for the interview, and this blogger is pretty hostile to you. But he cast about for some opinions or from some reputable scientists. The first one he got was from a guy named Richard Gibbs, who is one of the key scientists behind the Human Genome Project, director of human genome sequencing at Baylor College of Medicine. And he said, as a scientist, I would consider anything. The currency of scientific advance is keeping your skepticism at bay. You have to approach these things agnostically. As I read the paper, I asked, he says, is the evidence here compelling? He says, I don't know. Is there clear evidence of fraud? That is not apparent.
It's an intriguing hypothesis. One would need to view more sequencing information before supporting the conclusions. That's, I guess, about the best you could hope for. Is
that's
hey,
right. I mean, like I say, and that should have been a passing peer review, basically, because, when you do a paper, a lot of times there's going to be changes as you go further into it. We changed our hypothesis from the word go, because every time we kept testing something, we'd get some little nuance about it that we didn't expect. And so, all we're saying is we've got a novel hominid here. And obviously, by the placement on the tree and the placement, with the mitochondrial DNA, it either derived from human and mutated excessively, or it was a hybrid. And with the short time period between the birth of the, for lack of a better way of putting it, the birth of the mitochondrial type and today, it's such a short period of time, we believe it'd be very difficult for a true human to mutate that much between that time period and today. But conversely, and that's something they don't like, the established science, it's not exactly Darwinian. It's not exactly following Darwin's theory, and they don't like that. And I mean, that's what they've been taught, so that's what they believe. So yeah, they're gonna give it trouble.
Well, you can't blame them for that. I mean, that's a lot to chew on.
It is. It is. I mean, it's very unique, something that's not been seen before. And, we just, rocked their world, basically. And plus, they don't know me for the most part, because any recognition I've had has been in animal genetics and forensics. It's not been in human genetics. And these are the types of people looking at it. Who is this person that's come out of the blue with all of this? So, I can understand that, too. But, as scientists, the only thing I think is they should be fair. And, papers get published all the time that somebody will come out with a further version or an improved version of another paper as time goes on. All we were trying to accomplish with this paper was to show we seriously did everything in our power to make sure the samples were clean. We did everything in our power to species identify the samples, and we did everything in our power then to show that what we got was what we thought we had, which was a novel hominid, and the eyewitnesses and research programs linked it to the Sasquatch.
There was a guy from Princeton, a geneticist from Princeton, quoted in the same article that I just mentioned a moment ago. He says, no data or analyses are presented that in any way support the claim that their samples come from a new primate or human-primate hybrid. Instead, analyses either come back as 100% human or they fail in ways that suggest technical artifacts.
Well, that would be more like what the first paper was before we had the genome. Because if we had technical artifacts in the genomes, our quality scores would have been terrible. So there's no technical difficulties there. They're better than the average human genome that's run day to day for these big medical centers. So we know, and plus we had the histopathology done. We had the yield gels of the DNA to see how clean the bands were. We even degraded my blood and used it as a control. So that we could see what really degraded DNA looked like on these tests versus what we were getting result-wise. So
I'm sorry, go ahead.
Go ahead. No, that's fine.
What's been the reaction media-wise, the kind of coverage you've received so far?
They don't know what to do with it. It's been real quiet, eerily quiet. We got a lot more when we just put out the press release to counter the leak a month or so ago than we did this time. This time we have people asking for the paper, and it's just like dead silence. I don't think they know what to do with it because it is, it's unusual. It is. But, I mean, the data is what it is. Can't be altered the way we've done it. And we've used such good labs as well as our own labs, and all of it has backed itself up to the point that, if they would just judge the science and not what the results came out, and forget that it's Sasquatch, it would have passed. There was a monkey paper, LISTIA, published in PLOS One here recently. They got a new species on 6,800 bases of data. We have 2.7 million bases in one genome alone. And 6,800 versus 2.7 million, that's a huge amount of difference. Furthermore, we have whole mitochondrial genomes, 20 whole mitochondrial genomes and 10 partials that are, enough to give haplogroups. And they didn't even have that in the paper, not whole genomes at all, because the whole mitogenome, 16,500 bases, yet they published that paper. there's a new monkey in Africa with very flimsy data versus what we have.
What about reaction in the Bigfoot community? We talked about this the last time you were here, and it was all across the board. You had a lot of people who, as you just mentioned, regular people who had seen these things out in the woods or the mountains or something, and they said, good, I'm really glad to see some work being done on it. And then you've got people in the Bigfoot community who they have their own sort of sacred cows. They have their own theories that they want to support, and they don't like anything that's hostile to it. Since the paper's been out, what's it been like in that group?
Oh, the regular players have had a field day trying to find everything they can criticize. And, of course, they're not scientists, so a lot of the criticism is totally unfounded. They're just mad because it's not an ape, and they thought it would be an ape. And they don't like that here I am, a woman that's not part of the Bigfoot community has come out with this. So they're very vicious, extremely so. I've had threats and everything else, which is, very disheartening. I would have thought they would have been glad to finally have the mystery solved, but no, they're very unhappy about it. But once again, the majority of the people, I've had so many congratulatory notes I can't even keep up with answering them, and people so excited about it. The only thing I want to get done now is to make sure that overall the scientific community, there's going to be some that you won't convince no matter what. But we want people to know that it's out there, that it's legitimate science, and that it supports all these people that have had these sightings and all, and that they can come out now and talk about it without fear of being made fun of and what have you, or thought that they're crazy. I've seen them. I will admit it. I have seen them. 2 years ago it started, when I have seen them several times since. They exist. They're real. And, they're people. They are a type of people. We do not want them hunted. That's a terrible thing to hunt one, because it would be like hunting you or I. The other thing is, as I've said before, people are going to get killed in the woods, because if this doesn't come out into the media in a large way, people are going to, when they do find out about it, they're just going to panic and get scared, and they'll shoot anything that's on two legs coming toward their house. And that's not going to be good either. There's going to be people killed also. People need to understand that these beings don't want to bother us for the most part. They're peace-loving. They've lived under our noses for thousands of years. They're not going to hurt us. Just live your life normally. Don't be, looking for trouble when there is none. And, we've got studies underway in the background that are non-DNA studies about their culture and behavior. And, those will come out at some point. I doubt any journal is going to publish them, but we'll make them available somehow. And we're also starting a foundation for this type of non-invasive research, as well as to try to lobby to protect them. They don't need a lot of habitat set aside. It shouldn't really be a political issue. The only issue is protecting them from us, because we are their major enemy. And the other thing is, if we are trying to hunt them or hurt them, we don't know. They might retaliate back on us, and we don't want that either. We just want the status quo to continue. Let's leave them alone. Don't go in there and scare them in their own environment. Don't go in there and try to trick them and run them down. I mean, they're people. We should respect them like we respect each other. Of course, that's a lot to ask.
Yeah. I'm not sure we want to respect them as we respect other people, because
that's
you and I touched briefly the last time you were on the program about Native Americans, that there seemed to be some sort of a connection there on some level. And I would think it would be a mixed bag among Native indigenous peoples, because they seem to have a better understanding of Bigfoot. They've accepted them as being real for a long, long time, hundreds and hundreds of years. And yet you do hear stories among tribes about Bigfoot coming into a camp or a village and hauling women away, or people away.
There are a few of those stories, but by and large, the great majority of them, some of the people that are in my group have Native American or Indian heritage. They prefer to be called Indians, I've been informed. They have Indian heritage. I even have some Indian heritage, in which case most of them, they just cohabitate. They live and let live. Like I say, there's the occasional place here or there where there's been trouble in the past, but it's pretty rare. And most of the tribes they all have names for them, and that's many tribes. And most of the names are like, giant of the forest, or big older brother, or fairly friendly type names. Only just, 2 or 3 tribes have negative type names. So they've been accepted for a very long time. Our DNA shows that they were probably here before the Amerindians came over the Bering Strait. In fact, that's a criticism we had. But there's a very nice theory out, Salutrian theory, from a couple of Smithsonian PhDs. They wrote a paper and a book on it where they have found artifacts that indicate possible European settlement underneath the Clovis layer, which is, below the layers that are of the earth that are containing Amerindian artifacts. So there is a certain amount of scientific. It's another one of those theories where people don't necessarily want to accept it, but yet there seems to be good evidence for it. So we think that actually our findings might further support that.
Any reaction from Native Americans since the paper's been released?
It's been positive, very positive. They're not as outspoken about it. They have respect for them, as we should. And so they don't really get very vocal about it, but they want them left alone just like we want them, or, our group wants them left alone.
We put a link to your paper on our Twitter feed, Coast to Coast Twitter feed, but I should warn people there is a cost for it. There is a charge. What's the deal with that, and is there a point at which people will be able to read it without paying something for it?
At some point, we'll release it open access. Now, press, if they furnish us with a press pass, they can have it for free. People in the study can have it for free. But otherwise, we're trying to offset some of the expenses we had. It is common for most journals not to be open access. There's some open access journals, but your Science and Nature and a myriad of other journals do charge for you to upload an article. So this is accepted protocol. We're actually more reasonable than most of those articles. So, that's the whole point of it. Plus, our journal, the journal it was acquired to get this paper out, but it also is a real journal, and we've got a real editorial staff we've built in that we're going to be editing our website and adding. We've already had a couple of papers submitted, and so we've got, work coming in. What we're going to try to do with this journal is not that we're after anything cryptid. That's not the point. The point of this journal is to give publication alternatives for people that have manuscripts that have been shunned by the larger journals. And we're going to try to really watch the peer review to make sure that it's fair and not biased, because nobody should have to suffer this when they do good science.
Well, your hope would be, we got to go to a break in about 30 seconds, but your hope would be that, it does within a couple of years that this is a foregone conclusion, that it's no longer controversial, right?
Oh, it'll be before that, I'm sure, because it won't take that long for our results to be validated.
Dr. Ketchum, are you okay with taking a couple of questions from listeners?
Sure.
Okay, we're going to open up the phone lines with your questions for Dr. Melba Ketchum about her groundbreaking Bigfoot DNA study. Stay with us, everyone. Much more to come on Coast to Coast. Coming up in the second half of tonight's program, Dr. Lynn Ketai to talk about a very famous case, the Phoenix Lights case, which is a lot bigger and more extensive than you might imagine. For now, though, we're talking with Dr. Melba Ketchum about a fairly amazing and courageous study of Bigfoot DNA. Coming up, we open up the phone lines. I know a lot of you have questions out there. Let's hear them. Stay with us. Much more to come on Coast to Coast AM. My guest is Dr. Melba Ketchum, founder of DNA Diagnostics, talking about Bigfoot DNA. We're going west of the Rockies, Lori in Tuolumne, California. And I know that that's an area that used to be my stomping grounds, a lot of trees and hills, probably some Bigfoot sightings up there, right, Lori?
Oh, yes. There are a lot of Bigfoot sightings in this area.
What's your question?
My question was, considering that they're, archaic Homo sapiens, they're not us, but they're close enough to us. They're referred to as archaic Homo sapiens. And
I'm sorry, go ahead.
With the things I learned from reading the Earth Chronicles of Zachariah Sitchin, and how there was a primitive type being here, a primitive type humanoid, and they mixed their DNA with it. Is it possible that the Bigfoot, or big feet. I don't know what the correct term is for plural. Is it possible that they're the ones that the Anunnaki didn't get?
I have no comment on that because I'm a scientist. There's going to be a lot of theories that come out, but I can tell you they themselves are not archaic hominids. They are modern hominids because they didn't originate till 13,000 years ago or less. They're not ancient at all. Now, what sired them originally, the male fragment, we don't know where it came from or how long ago it was, but it's where it's located on the Tree of Life. We just can't tell that. It's not close to any of the apes or anything else, so we don't know what it is. But the actual Bigfoot as they exist today, the Sasquatch, are not ancient at all.
Well, I guess it depends on what you mean by ancient. Is that
they're not archaic? They're not ancient. They're extant. That means they're alive today. They originated, not very long ago, and so normally when you think of your archaic, you're looking at your much more ancient fossil peoples of like the Denisovan and the Neanderthal, and they're further away and further removed from where we are. Now, that's not to say that the progenitor, the one that or the original father, you might say, to the Bigfoot species, we don't know where they came from. We don't know how old it is. We don't have any. At this point, we don't have any DNA that could have come from that. So we don't know what it is. So there's going to be all types of speculation. All we're trying to prove with our paper here, or we have proven, absolutely, really, without a shadow of a doubt, is that we have a species originating from modern human females that is somewhere less than 13,000 years old in some of the samples, and therefore they are a modern extant species. They're still alive today, and they are considered modern because they're in parallel with modern humans.
Interesting idea, though, Lori. Thanks for the call. Appreciate it. Wildcard line, Nancy in Oregon. Hi, Nancy.
Oh, hi. Thank you, George. Great show. And I want to thank Dr. Ketchum for her perseverance. It's just incredible what she's had to put up with on this. My question is there enough DNA data to indicate how much phenotypic variation there would be among the Sasquatch or the Bigfoot, whatever we're calling them? Because sometimes you see conical heads and sometimes you see rounded heads and different types of facial features that look more human or less human. And I'll take my answer off the line, but what would it take in order to get that kind of data? Thank you.
Interesting. Thanks, Nancy.
Okay, we already have that data. It's just a matter of mining it out of the genome. We do know that there's a lot of individual variation, not only from eyewitness accounts, but we even are to the point that we can't prove this yet because we have to go further into the genomes, but we think we have probably 3 different variants represented from 3 different parts of the country in our genome. So, when we get through the genomes in the next, few years, we'll know a lot more. But we do know that one of the samples did not align as well. We think it's a little more primitive. It came from South Alabama. So with that in mind, those were the so-called skunk apes, which is allegedly a different variant. And eyewitness reports say that it's more primitive and smaller. And with it not aligning quite as well, we think that possibly we have a skunk ape there. That's not something that we can, we're sure enough of to put in a scientific paper yet, but it looks that way.
Nancy raises an interesting point, though, Melba. I mean, some of the artist sketches, the forensic sketches, show a lot of differences in these things.
Oh, absolutely. And, like I say, I've got, pictures of several of different ones, and they're as different as you and I, look how much variation there is just in the human race. I mean, you have all these different races, and each one has very much physical differences as far as appearance. And you can, and when you do a little forensic anthropology, even you see differences in the skulls of the different peoples of the world. Well, you're seeing something similar here. I really believe there's as many different appearances or phenotypes of Sasquatch as there are, peoples of the world, regular Homo sapiens.
Yeah, I can only imagine an alien coming here and looking at the different kinds of humans, figuring there must be all kinds of different species. Jeff in Simi Valley on the wild card line. Hi, Jeff.
Hi, great to talk to you guys. Excuse me. My question is kind of what you're alluding to right there. I was wondering, I believe that Bigfoot and these are all for real, and I was wondering if you suspect that there are different races from different places around the world, just like, we've got whites, blacks, Orientals, if they are kind of, on the same order as that they develop different around different places of the world.
I believe that's probably true because of the different reports over the globe of little bit different phenotypes. Like I say, even just in this country with the skunk ape versus like the western Sasquatch or eastern Sasquatch. And it seems the western Sasquatch appears more human-like and much taller, and the eastern one's a little smaller, just slightly less human-looking. And then the southern one looks much more like an ape. So, with that said, I believe that there will be around the world. I think, though, that one thing they're going to have in common probably is the human maternal lineage, because we've already played with a couple of samples out of Russia. We got one of them we've already got some data on, and it looks like it's going to line up with what we got already as far as the human mitochondrial DNA.
Thanks, Jeff. Mike in South Florida, down there in skunk ape territory. Mike, what's on your mind?
Hey, guys, how are you?
Okay.
Okay, I don't like to call the animal or the creature Sasquatch. I think it sounds like a vegetable. I like to call it a Bigfoot. It gives that creature justice. Bigfoot sounds more reasonable. I got to say something. You have bears that have the same fur and physical physique and that Sasquatch. But yet, every time you hear the Sasquatch, you hear about that smell, that same odor. So bears that hibernate for a certain amount of time, they burrow under the ground, they don't have that type of odor. So I say the Sasquatch is a very expert at burrowing underground and hibernating for a long time. What Sasquatch does better than any other animal, it's very easy to disappear and never seen again. And so when you bury yourself under the ground somewhere damp, you have a tendency to hibernate and have that odor, obviously longer than a bear hibernates. So I like to say the Sasquatch is very expert at burying themselves. And you know what? They don't want to be seen intentionally. They come up to feed and they bury themselves longer. And I think this needs to be investigated, or maybe some scientists need to see that maybe underground something that they go under and they bury themselves. So that's why they have that odor, because hibernating for a long period of time.
Well, let's ask Dr. Ketchum about that, Mike. Dr. Ketchum, any evidence that they are burrowing and hide in that way?
Not that I've seen. When I've been around habituation sites, they're pretty well out most of the time. Not to say that they don't live in. I know they live in caves if they're available, and I know they're going to find protection for themselves one way or another. But as far as any proof of burrowing per se, that doesn't seem to be the common trend. As far as the odor, to me, I think it's more of a BO with a little tincture of odor skunk on top of it. I've smelled them a lot of times, and it's a very unique odor. Once you smell it, you will always recognize it.
Glenn in Tampa on the wild card line. Glenn, how you doing?
How you doing? I was going to ask your guest here. I listened to David Politis, your other guest you've had a couple of times, and Coast to Coast, and he always said that there's concentrations of these type of creatures. And I was wondering, is it like that on what you've seen as evidence, Doctor, or is it more, is there isolations, or is it more like an even pattern all around the USA of their existence?
They're more common in forested areas. Obviously, it's easier for them to blend in. But there's been sightings in all 50 states and all the provinces in Canada. The only place that has not had a sighting is Hawaii, for probably obvious reasons. But they're not, as long as there's brush or some kind of cover, you will find populations of them. There's a lot more of them out there than people think. I mean, the most I saw at one time during a habituation visit was 9. There were 8 in front of us and 1 behind us. And we were able to count them in the shadows quite clearly.
Glenn, thanks for the call. East of the Rockies, Lloyd in Lafayette, Louisiana. Hi, Lloyd.
Good evening, George, or good morning here. And Dr. Ketchum, wonderful program, great information. My question is, well, I don't know. Let me ask. Have you found any or had any skeletal remains to work with as far as DNA?
We're working on some.
And if not, why has there not been any fossil remains found, in your opinion?
Well, they're a type of people, and I believe they have intelligence, and they probably bury their dead. That's the indication we're getting. Now, as far as we do have some skeletal remains that we're working on right now, if we can get good DNA from them, this may answer some more questions. But it takes longer to do that, and there's less available DNA. So in order to get the study finished, we use, soft tissue sources first.
Appreciate it, Lloyd. West of the Rockies, John in Seattle. Hi, John.
Hi. So my question is about the research that you're doing as far as their culture. I would be interested in anything about that. I'm a part of the BFRO up here in Washington, and actually saw a Sasquatch last year, so still kind of blessed to have seen one.
Oh.
Well, I tell you, I might just put it out there. Any habituators that are out there might give us a shout on our media at SasquatchGenomeProject.org email, and because what we're doing is having them independently basically put down the different interactions and experiences. Nobody knows what the other one's doing or who they are or where they're from, and then we're going to take these studies and compile them with what is in common between people, all these different people that have interaction with them. I didn't believe it was so until, I became basically a habituator. I have a site where they are, and they've come out, and so it's going to be very interesting, all of the different reports. But over the years of us having the study, a lot of people have contacted me, and I've talked to a lot of people, and what just blows my mind is that you'll have somebody, say, on the East Coast that doesn't know somebody on the West Coast, and they'll have the absolute same story. They don't even know who they are. They may or may not even be active in the BigFoot community, and yet they've heard about the study. They call, and we've already just learned volumes about this, and we want to compile this to help, move forward, because these are people. They're a type of people. They are not animals. They don't need to be harassed, made fun of, made noise at, shot at, chased. Don't go curiosity looking in the woods. You're liable to get shot by another person at this rate.
John, John, I appreciate the call. Maybe you should send a note to some details to Dr. Ketchum about your sighting, and it makes sense, Melba, to have as big a database as you can collect, I guess.
Yes, and that's what we're doing, because these are all the $64,000 questions. Well, why don't we see them all the time? Why don't I've got answers to a lot of these things that at this point we're not going public with, but it will be a huge study with a lot of people involved and wonderful information, because so much of it lines up from one individual to the next, which is great.
A first-time caller, Joaquin in El Paso, Texas. Hi, Joaquin.
Oh, hey. Hi, Dr. Ketchum. I just want to let you know, first off, I've been following your work for a while, and others like you, like, for instance, Ash and them. But I wanted to ask, when you were getting the samples, I would think, I don't want to take every chance I could to get evidence of a Bigfoot or a Sasquatch. So why didn't you try using something like a game cam? Because I would think, I got to catch them all. I got to get all these pictures I can.
They avoid cameras like the plague. They know they are there. In fact, animals know they were there, because we put some out at my lease where there are Sasquatch, and I noticed my horses would obviously know the camera was there and approach it and look at it. So if the horses know it's there, I know good and well that the Sasquatch know it and avoid it. And this is across the board. They just avoid having any contact with electronics. So, actually one night we were out, and I didn't see them there, and I was playing with a new spotlight, and I started to pull the trigger on, and I saw this brilliant red eye shine, like during the headlights look, and I couldn't get it stopped, but I didn't get it right at them. I tried to lower it, and immediately, of course, they disappeared, and then about, 20, 30 feet down, then I saw the eye shine again, and then it was the more normal, pale, whitish green, where they had moved down, but obviously I frightened them by aiming anything at them. So, it's definitely, they know what's going on. They know, an animal can read you and feel you. We'll take that and add human intelligence that's used to living in the woods to it. The other analogy I give is like a special forces soldier. If he's in full camo and you walk through the woods and he's hidden, you're probably never going to know he's there. Same thing with the Sasquatch. They know how to blend in better than any creature on the planet. So, that's the main reason you don't see them.
That's a good point, though, Joaquin. And I guess you do have some isolated images, though, Melba, and you're going to be talking about that more in the future.
But once again, that was almost to the point that they had somewhat, domesticated in the right word, but they kind of made friends with them to the point they were able to get this. That is the exception rather than the rule, absolutely.
Well, we appreciate you coming on to talk about the study. Hope you will keep us in the loop. I mean, obviously, this is the beginning, just the beginning still. You've been at it a long time, but it's still just the beginning of a great adventure.
It really is, and I really appreciate you having me, George.
But, I mean, you think this is a solvable mystery, though?
It's already solved. It is absolutely scientifically already solved. It's just getting mainstream science to come up to speed, and that's starting to happen already. I've had several requests by people that are well known, to look at the data, and that's fine, that's good. We're happy with who's looking at it. I'm anxious to hear their feedback on it, but like I say, we got 2 passing peer reviews that and they were critical of certain things and what have you. You read them, but they passed it. So that means, and they were genomic specialists according to the editors. So we've passed muster already. Sasquatch does exist.
All right. Dr. Ketchum, thanks very much. Appreciate it, and hope you'll come back and talk to us again sometime.
We will. Thank you so much, George.
All right. Stay with us, everyone. Much more to come on Coast to Coast. Dr. Lynn Ketai to talk about the Phoenix Lights, and there's a lot more to it than you might imagine. Stay with us, everyone.
Monster.